Hello and welcome to this video, and in this video we're going to be discussing the topic of protein synthesis as one of the main processes that the cell actually carries out in order to do everything that it's intended to do, growing up, developing, having balance within its metabolism. Everything that the cell does is mostly focused in this process, which is something that we call protein synthesis. The purpose of this video is to review these concepts and to understand the general aspects that make up this particular process that includes two steps. So as a whole, protein synthesis, we can simplify it in step number 1, which is transcription, and step number 2, which is translation. So the purpose of this video is to have it in a very simplified way, what is going on in transcription and what is going on in translation, and how does this actually have an effect into what the final product is of this process, which is at the end is to make protein. So synthesis is a word that means making, and protein is one of the 4 biomolecules that we started studying early in this semester. You may recall when we talked about the 4 biomolecules, we mentioned the carbohydrate, we mentioned the lipids, we mentioned the proteins, and the nucleic acids. So this chapter is pretty much the relationship that exists between nucleic acids and proteins. One is going to be responsible of creating the other one. And you may recall from previous chapters that we have the 2 greatest examples of nucleic acids, which will be DNA and RNA, right? So the Na in RNA and the Na in DNA stand for nucleic acid, right? So DNA, RNA, and protein, these 3 components have a very close relationship, and this topic is the one where we will explore this relationship between DNA, RNA, the 3 types of RNA, by the way, and protein as the final product of this process of making protein. Now, you may recall also from previous chapters or previous topics that we were talking about the different parts of the cell that include certain organelles, certain structures, such as the ribosomes. These ribosomes here in this animation are these blue things that look like a burger that open and close, and those little worms that come out from this big structure to the left is one type of RNA. So if you may recall, the nucleus, which is this big thing in the left, the nucleus is one of the largest type of organelles that we have in the cell, and you may recall the function of the nucleus is to store genetic information. Which genetic information? DNA, right? So let's put it in here. Wait, I'm covering it. So the DNA, which is located in the nucleus itself, is made up from its monomer, right? So nucleic acid has a monomer that is made, or that is called the nucleotide, right? And this nucleotide itself is made up of smaller units that are called the nitrogen bases. And you may recall from previous chapters, the nitrogen bases are a total of 5: adenine, thymine, guanine, cytosine, and uracil. 4 are specific to DNA, and 4 are specific to RNA, right? So nitrogen bases will come together with the pentose sugar, the phosphate group, in order to make this nucleotide. You may remember this as a drawing that includes like a pentagon, a rectangle, and a circle, where you would have on the left side the circle representing the phosphate group, the pentagon will be representing the pentose sugar, and the rectangle the nitrogen base. That would be like the monomer. Monomers come together to make polymers. In this case is DNA. And DNA, big chunks of DNA at least, is what we call the gene, right? Which genes are located in chromosomes, and chromosomes within or make up the total of your genome. So in this chapter, basically what we study is how these genes that are made up of DNA will be responsible to create copies of this information in order for these working guys, the ribosomes, which, by the way, these that you see here are the free ribosomes, meaning they're free in the cytoplasm, not attached to an organelle. But we can also have the rough endoplasmic reticulum with those ribosomes attached to it. So either free ribosomes or rough endoplasmic reticulum, which contain ribosomes, by the way, will be the ones responsible to convert this genetic information into the final product, which is the protein, right? In previous chapters, we have also mentioned when we were studying heredity or inheritance that genes are responsible of making proteins, right? So let me just complete here. Gene, the chromosome, and oops, what's going on? Well, chromosome and the genome, right? So from genes, we actually can have a specific code to make what we call the protein. So this relationship between DNA, gene, protein is today's mission. Now, another thing to remember is when we talk about proteins, just as for nucleic acid, we have the monomer, which is nucleotide. For protein, the monomer of protein, you may recall, being the amino acid. So when we're building proteins, we're going to be using the building blocks, the original monomers, to make protein. So every time that we talk about making protein or synthesizing protein, we're going to be talking about amino acids. Now, for our course, it's not required for you to memorize the amino acid names or the categories of the amino acids, as long as you understand that there's 20 different amino acids, right? So 20 different amino acids are going to be, let's say, available to have a lot of different mixtures and combinations among them in order to start building protein as a larger unit of a molecule that we call a polymer or a biomolecule, which is the same thing. Now, this doesn't mean that there's only 20 amino acids available and the largest protein is 20 amino acids large. It just means that there's different versions. Let's say, imagine it's crayons, right? Or different colors. So you would have a different types of colors, but you can make a lot of different combinations using those 20 colors. So the point in here that I want to make is that everything that is alive, everything that has life, it's actually made up using these 20 different versions of amino acids into different types, quantities, shapes, forms of proteins that will be making different functions within your body. Now, something else that is important to remember when we were talking about the different nucleic acids, such as DNA and RNA, is the 3 different types of RNA that we would have. We have the mRNA, the rRNA, and the tRNA, right? So M in mRNA stands for the messenger, which will be sending a message. T is for transport or transfer of the amino acid from one place to another one. It will be like the ride for the amino acid from one place to the other. And the rRNA, this R stands for ribosome. So this ribosome, or the organelle that is in charge of protein synthesis, will be using a big chunk of this information. So when we talk about protein synthesis, we're talking about three major players and two processes. The whole process starts with DNA. It will continue with mRNA, by the way, and the final product will be protein. So this process of having one DNA and making a copy of the DNA to make mRNA is something that we call transcription, which is making a copy of DNA into a structure that we call the mRNA. Going from mRNA and actually getting the final product out of this code is what we call translation. And this will have a specific place for this to happen, which coincidentally would be the ribosome, right? Because at the end, the ribosome will be the one in charge making the protein. So we can start to make connections as using this terminology, where the ribosome will be the one in charge of making translation. Now, using this image, we can also start to identify also how to make the connection between the processes, the players, and the location. So within the nucleus, where we have the DNA, or big chunks of DNA that we call a gene, this is where we will have the copying, or the making the copy of the gene, which is what we call transcription. After having the mRNA constructed, this mRNA will exit the nucleus through a nucleus pore and coming out into the cytoplasm, where it will meet the ribosome, which is the organelle in charge of making the protein, and it will join forces. This mRNA will join forces with the ribosome and will start to mass produce or chain produce amino acids joining together, so we can start making a very long chain of amino acids. And as we start to put amino acid next to amino acid, we will start making a very large protein that will be having a particular function in or outside of the cell. A good metaphor or analogy for what's going on in here is that the cell being like a factory, and this factory has a chief, a leader, that is the decision maker. Now, this person in particular has all the information available, has all the different plans and the different resources, information in order to make a decision. But not everything will happen always at the same time. So depending on what the leader actually decides, what is best for the moment, this person will make a decision. This person would say, okay, you know what, we need to actually right now at this moment start to think about what is coming next, and according to our needs, this is the thing that is coming up next. So in this analogy, this leader would be the DNA, which will provide an instruction to a supervisor, let's say, which is represented by the mRNA. Now, this mRNA will actually take the hint and will say, okay, let's do it. So whatever it is that you want, we'll take it out and into the main factory production area, where this mRNA will provide the message or the copy of the message that was being told by the CEO or by the leader into the working guys that will be responsible to manually and individually, each of them, make the final product, which would be a protein. So looking specifically for transcription, what can we say about transcription? Transcription would be the process that occurs within the nucleus, right? So we'll be taking inside of the nucleus, and basically we would be taking the DNA helix opened again, like when we happen in what happened in DNA replication, so we can actually use the information from the DNA to make a complementary copy of the information that we have there in the DNA. So if we have, let me put it in here, if we have this DNA being like a double helix originally, and then we split it open temporarily, and then this area just comes back to the double helix, then we have this open space where we can, by unzipping the double-stranded DNA, we can take a little bit of area from this to take as reference the information that is present in there. Let's put the nitrogen bases. Let's say, I'm going to put it here, A, C, G, C, G, A, for example, right? So we're going to open this strand, and we're going to use this strand as reference. And we're going to start building a copy based on the complementary bases that we have available in there. So if we have A on this strand, then on the other one, we would have U. Now, why U and not T? Because in here, what we're making or what we're building is RNA. So we cannot have the T or thymine within our RNA. So we would replace it by U. And the other pairings are going to be the same. So with C, we pair G. With G, we pair C, CG, GC. And with A, another U, right? So we're going to start building a complementary strand based on original information that we have there. But this strand is not staying there forever. We're going to build it, and this strand will be exiting the cell next, and continue the process to go to the ribosome in order to follow the second step, which will be translation. But so far here inside the nucleus, we're still in the nucleus, right? So far here in the nucleus, the enzyme, or the one that is actually going to build the mRNA, is an enzyme that is called RNA polymerase. The name itself is telling you what it's supposed to be making. You may recall before we said that ASE, this ending, ASE means enzyme. Polymer is a large molecule. And the first part is telling you molecule. And the first part is telling you which molecule in particular, right? So this RNA polymerase literally means an enzyme that will make a molecule that is called RNA. And in this particular case is mRNA, right? Because that's the whole thing that we're trying to do here at this stage in transcription. So in this diagram, you can see the same thing where we have the blue one. The blue strands are the DNA. You notice how there's two. There's complementary, right? And they're in a double helix. All of these are characteristics of DNA. And the red one is the RNA. Which one specifically? mRNA, right? This is the messenger. This, how do you know it's mRNA? Only the red one is single-stranded. It contains uracil, different than the other one. Notice how it contains the thymine, and it doesn't contain the double helix. So at this point, you may be confused about what's going on in here. Why is there a double-stranded, right? But the thing is, in here, in this diagram, what we're looking at is the process of actually building RNA. And you may recall that this enzyme, RNA polymerase, which is represented by the green thing that you see here in the background, is the enzyme responsible of attaching nucleotides, complementary nucleotides, according to the original strand that it was unzipped and unwind in this stage of DNA, so we could use it as a reference to make the new strand. So in here, for example, this is like all of this is the mRNA, right? So in this stage is how these nucleotides are actually being set in here with the complementary strand, this one with this one, this one with this one, and this one with this one. Notice how A now joins with U, but this is temporary. It's not staying there forever. This is just so that mRNA can actually follow the same code that is embedded in the DNA, and eventually this RNA will exit the cell and continue to the second stage of the protein synthesis process. Now, the second stage is called translation. This is the second part of protein synthesis. This occurs in the ribosomes, either the free ribosomes or the attached ribosomes, specifically to the rough endoplasmic reticulum, or the free ribosomes that are free, meaning free in the cytoplasm. Either of this is a ribosome, either of this, their function is protein synthesis, and either of this are going to make exactly the same thing. What is this happening, or what is this actually doing? So we would have, number one, mRNA present, which is this worm that you see here, right? Notice how we have the nitrogen bases sticking out, right? CGU, GAC. Now in here it might not be as evident, but notice how they are organized in small groups of three. This, for example, these first three are one group, then these are another group, these are another group, and so on, right? Each of these groups is called a codon. And by definition, one codon is a group of three nitrogen bases that are going to be exclusively present in the mRNA. So if I ask you, where is the codon? You would say, in the mRNA, right? Now, what is a codon? This word comes from the word code, right? Or it's trying to make a reference, at least I think, to the word code, in the sense that each of these, like a barcode, as in a product that where you pass it through a scanner, right? It will have a specific code for a specific product. Now in here, this codon will be specific to one product. And what do we call this product in here? One amino acid. Now you may recall before we mentioned tRNA. And remember that the T stands for the transport of amino acids. Where to? To the ribosome. What for? Translation. Why? Because this amino acid, being attached to this T-looking structure, T for tRNA, is going to have an anticodon. What is this anticodon? It's also a code that, if you notice in here, is complementary to these nitrogen bases. So overall, the codon will be a code of three nucleotides that will match exactly with the anticodon that is present in the tRNA that is specific for each amino acid. In other words, depending the codon, depending the amino acid, right? So that's what we mean when we say that the tRNA has as a function to transport the amino acid specifically for translation. Because in this section, what we're doing is, you may notice here in the background, there's a green thing. This green thing is the ribosome, which is representing the organelle where all of this is actually getting together. So in summary, the ribosome joins with the mRNA, and depending on the codon, the anticodon of the tRNA will match, and because the tRNA transports the amino acid, this will start to have a chain reaction as as the ribosome, like a train. It will behave like a train. It will continue to advance forward from the mRNA, so more and more amino acids start to form. How exactly? Well, because each codon, this is amino acid 1, then it will move forward, and amino acid 2, move forward, amino acid 3. You notice how there's 3 tRNAs here? Because it moves forward, amino acid 4, and then it continues to move forward, amino acid 5, and then amino acid 6, and then amino acid 7, until there's thousands of them. And the final result will be the protein. Now, let me show you an animation on how this is actually happening. Now, looking at this simulator, in here we have the original 2 strands of DNA. How do we know that this is DNA? Because we have the thymine in here, 2 strands, and they are bonded by hydrogen bonds. If we click here, prepare for transcription, notice how the 2 strands separate, and we can actually start doing the complementary nucleotide to start building our own RNA, specifically mRNA. So T binds with A, and A with not T, because we're making RNA, so it has to be the U. C with G, A with U, U, and then G, and then A, and so on and so forth, right? So we continue making this mRNA that eventually will exit the nucleus and go into the ribosome. And if we look at the next simulator, which is for translation, in here in translation, we have the yellow thing, which is representing a ribosome. We have the red chain in here, which is representing the mRNA. And what happens if we click in here in translation to begin? We will have these green things approaching, which the green thing is representing the tRNA. So notice what's going on. The ribosome is moving forward like a train. And the green thing that is representing the tRNA carries one individual bubble that represents the amino acid. And how is it chosen which amino acid? Well, because in the bottom, connected to the mRNA, this is where we have the codon, which will attract one specific amino acid. And as these moves forward as a train, these amino acid plus amino acid plus amino acid will continue to make a very long chain of amino acids, which is called protein. So in summary, in here we have the transcription process basically going from DNA to mRNA, and from mRNA to protein is the process of translation. Both of these together make the process of protein synthesis, right? Where does transcription occur? Occurs within the nucleus, right? Because this is where DNA is located. As soon as transcription is finished, this single-stranded genetic information, which is a copy of the DNA, by the way, will exit through the nuclear pore, arrive to the ribosome, and the ribosome will start reading it, and the protein will be produced and exit as a result of the translation process. All right, so that finishes pretty much the explanation for this video. I encourage you to take your notes, watch the simulators. The simulators are available in Canvas as well. Or you can personally ask me for more details, questions, or for further explanations in case you may need it. Thank you for watching, and I'll see you around.